So next I want to talk about mitosis. So we've discussed about all the preparation for the process of the cell actually undergoing the reproductive aspect, the splitting, and the requirements that are necessary for this. The cell being of the right size, the right amount of energy available, nutrients available. Are there growth factors, the mitogens present, to stimulate and regulate this process? Is the DNA completely replicated? And like I said, once we've met all those requirements, then the cell can go from the checkpoint, G2 into the M phase. Okay. Now, basically, what you're going to see here is kind of a systematic way of the cells dividing and making a copy, an identical genetic copy of itself. All right. First, we start out in prophase. All right. Prophase. Now, basically, during prophase, what you're going to see is the genetic material in the nucleus condense into chromosomes. So all that replicated DNA is going to start to condense and form these very dark-looking lines within the nucleus called chromosomes. All right, and you're going to start to see them congregate within, around, and like I said, this happens in the nucleus. And this is just kind of a zoom in of what it looks like. You kind of see these elongated bulbous ends and a very thin center with something called a kinetic core in the center of that chromosome. And we're going to come back to that in a little bit because that's important. So now also what you characteristically see in prophase is, so you start to see the development of the chromosomes within the nucleus, and you also see the nucleus start to fall apart. The nucleus starts to degenerate and fall apart. And again, you still see a little bit of it around, this genetic material itself. But for the most part, the nuclear envelope is starting to come apart. All right, and then also what you see are these structures called centrosomes. They basically start to migrate towards the poles of the cell itself. When I say pole, I mean they're just going to opposite ends. Okay, I have them drawn in on these ends. I mean, they could be up here, they could be at any angle. Just understand that they're going to migrate to opposite ends. So that's characteristic of prophase. Again, you start to see the DNA condense into chromosomes, the nucleus starts to fall apart, and then you see the centrosomes migrate towards the poles of the cell. All right, and then there's a very quick little stage that happens next called prometaphase. All right, and then basically what happens during prometaphase are these chunks of the nucleus basically break off and then expose the chromosomes within the cell itself. And then you'll notice that the centrosomes are now basically at the poles, and these mitotic spindles are attaching to the center aspect of the chromosomes called the kinetochores. All right, now that's important because, like I said, these are going to attach to the centers of these, and that's going to play a role in the cell division in a little bit. Okay, so that's just a quick little phase you see there. And then next, you'll see a transition into what's called metaphase. Okay, and during metaphase, the nucleus is gone. Okay, the nucleus is completely gone, and now you also see again the centrosomes are at the poles. All right, now what's unique about metaphase is how the genetic material, how the chromosomes align themselves. Okay, they align along what we call the equator of the cell. All right, the center of the cell. Okay, and also as they align, the centrosomes at each pole, okay, they attach on each side. Okay, so before we can transition from metaphase to anaphase, there's two things that have to happen. One, all the chromosomes. Now remember, there's 42. There's 42 chromosomes here. In this process. So basically, but I only drew a few in there. So all the chromosomes are aligning, and then what you notice again are the microtubules of these centrosomes are going to align in those kinetochores. Okay, and they're going to attach on each side. There's going to be a little bit of overlap on them, but again, all these microtubules need to be attached to the chromosomes as well. All right, and then once this happens, once the alignment takes place and the attachment takes place, then the cell is going to transition into anaphase. And basically what you see occurring during anaphase are the microtubules are going to pull, they're going to pull these chromosomes apart. Okay, they're going to pull them apart into these into this daughter genetic material. All right, And these chromosomes are going to be pulled towards each pole of the cell itself. So that's basically anaphase, the separating of these chromosomes. All right. And then what will happen next is you work your way down to telophase, and well, you see a couple of things happen here. One are you see the chromosomes are pulled towards the poles. Okay, so basically they're pulled towards the each end of the cell, and you start to see the redevelopment of the nucleus on each side. So what you're looking at here is the formation of you're looking at the beginning of the formation of the new daughter cells. All right, and then also what you'll start to see is an invagination along the equator of this cell and of the membrane, and you form what are called contracting rings. Okay, and then within the contracting rings, you see these arrangements of actin and myosin, the contractile proteins that you would find in muscle. And basically, what's going to happen once we transition from telophase into the next stage called cytokinesis, how this exactly happens is not well understood. But these proteins are going to contract, and they're going to basically start to separate the two daughter cells from one another. Okay, cyto meaning cell, kinesis meaning movement. Okay, so again, these contractile proteins are going to start to compress, and again, how exactly is not well understood, but they're going to compress and separate into the two daughter cells. And then again, as you can see the nucleus redeveloping, around each set of daughter genetic material. And then once cytokinesis is finished, then you have two genetically identical copies of this cell, of what we started with, of the daughter cell. All right, now another thing to keep in mind as well is that organelles, the equal amount of the organelles from the mother cell has to be transferred and packaged over to the daughter cell as well. This isn't just the transfer of DNA and genetic material. All the parts of this daughter cell, of the mother cell, are moving into the two daughter cells. All right, but the main focus of this talk is obviously along the genetic material because because basically, remember, DNA is what drives the activity of the cell. The protein synthesis activities of the cell. So as a result, when we take a look at each of these steps, the genetic material has to condense itself just right into the chromosome. That's separate and condense into 50-50 portions. Okay, like I said, not only do the chromosomes have to align along the equator, but there has to be attachment of these microtubules to each individual kinetochore on each side. So while they're doing the pushing and pulling aspect of this, they're pulling in equal amount, 50-50, percent ratio of genetic material to each new daughter cell. All right, and then we go from there. So basically, what you're looking at here, like I said, if you watch this process under a microscope, these are the physical characteristics you're going to see happening. Again, with prophase, you have the nucleus starts to fall apart. You see the chromosomes, and you see the centrosomes moving towards the poles. And then during prometaphase, the nucleus kind of spreads away from the condensing chromosomes in the center, and these mitotic spindles are now at the poles. And then metaphase, you see the chromosomes align along the equator, and the mitotic spindles are attaching to each kinetochore, the center aspect of each chromosome. And then during anaphase, the spindles are pulling apart the chromosomes. During telophase, the genetic material, the chromosomes are now on each pole, and you start to see the beginning of the physical division of the cell. And during cytokinesis is the physical splitting itself. So basically, these first stages are just all about transferring the genetic material into each daughter cell, and also the organelles as well. And then once all this is transferred, then cytokinesis can occur, and the cell can physically split into the two daughter cells. All right, I know that was kind of a quick explanation, but I hope that kind of helped bring some light to this process. Again, I would recommend watching, before watching this video, go, watch what I discussed earlier about, interphase and the overview of the cell cycle, the overview of cellular reproduction. Watch, the requirements for cellular reproduction as well, because that'll just make this make a lot more sense. understanding, you understand the why